everyone. Today I'll tell you about circulating tumor cells, which are essentially tumor cells that are shed from the tumor, primary tumor, metastatic tumor, into the circulation en route to forming metastases. These are extremely rare cells, but the isolation of these cells do offer the opportunity for analysis, both before treatment and as well as longitudinal analyses during treatment, essentially acting as a potential liquid biopsy. And the issue, though, is that these are extremely rare cells, estimated at about 1 in a billion, and it's a huge technical challenge to isolate them. And I've had the privilege of working together with the MGH circulating tumor cell group here, led by Mehmet Toner and Daniel Haber. And this team, over the years, has developed a series of microfluidic devices that are able to isolate these cells quite effectively. This is the most recent version of the isolation device called the CTC-iChip. I stands for inertial focusing device. And the advantage of this device is that you're able to isolate untagged CTCs from whole blood, and this is done through microfluidic depletion of normal hematopoietic cells from the whole blood. So starting with whole blood, we tag them with magnetic beads that are coated with anti-CD45 antibody. This goes through hydrodynamic cell sorting and inertial focusing. And finally, we end up with white blood cells in one tube and an enriched population of CTCs in another tube. And this is in contrast to some of the other CTC isolation technologies out there, which rely on positive selection, where you have to know the markers ahead of time. And the CTCs in the end get coated with all these magnetic beads, which often interfere with downstream assays. So our team at MGH is working with Sony as well as Janssen to mass produce it, and we've repurposed. Sony has repurposed compact discs which are no longer being sold in an attempt to mass produce this. So looking at prostate cancer, which is my clinical interest, we've been working to develop, to look at how CTCs can inform treatment decisions, as well as try to understand potential mechanisms of resistance. And to just focus, the most recent FDA-approved treatments of metastatic castration-resistant disease are listed here. And just to focus on abiraterone and enzalutamide, which are actually targeting the androgen receptor, the abiraterone through steroid synthesis, enzalutamide through direct antagonism. And can we use CTCs, number one, as a biomarker to inform whether or not such treatments are working or not? And also, can we use CTCs to dissect potential mechanisms of resistance? So first, in terms of biomarker work, we looked at CTCs, and not just looking at enumeration, but can we actually measure AR activity in CTCs? And what we developed was this assay that depended upon a gene which is upregulated when androgen receptor is turned on, PSA, and a gene that's upregulated when androgen receptor is turned off, PSMA. This has been shown by a few different groups, including ours. And if you label PSA in one color and PSMA in another color, you can see a shift in the color of the cells as you go from an AR off state to an AR on state, and these LNCAP cells that are stimulated using an androgen. So when we applied this to CTCs, we initially looked at castration-sensitive patients, or patients with newly diagnosed metastatic prostate cancer who were treated, who were being initiated on androgen deprivation therapy. And pre-treatment, you see, as expected, that most of the cells have high PSA expression, but very low PSMA expression, reflecting their AR on phenotype. And when you treat these with when you initiate androgen deprivation therapy, you get this emergence of this PSMA population, which reflects, again, this AR off phenotype. So this is exactly as we had expected. When we, in turn, looked at castration-resistant prostate cancer cells, we found this emergence of striking heterogeneity. So again, in castration-sensitive, most of these cells are PSA positive, PSMA negative, but in castration-resistant prostate cancer, we often saw a tremendous mix of different AR phenotypes. So some of these were AR on, some of these are AR off, with high PSMA but low PSA. Others had this AR mixed phenotype. And when we looked across multiple different patients, again, most of these castration-sensitive patients had high PSA expression, low PSMA expression, but castration-resistant patients had this tremendous diversity and heterogeneity in their CTC phenotypes. Some of these did correlate with prognosis. I'm not going to get into that here in the interest of time, but I'd like to really focus on this heterogeneity issue, which is really interesting to me because it just suggests that as patients go through treatment, we get the emergence of these multiple different phenotypes, really reflecting increasing heterogeneity as patients develop treatment resistance. And now, to dissect this further, we decided to take an approach of RNA sequencing, so not just looking at two markers, PSA and PSMA, but what are the other pathways, genes that are differentially regulated in these cells. And so we decided to perform RNA sequencing of single prostate CTCs with the idea that sequencing these may uncover cellular pathways that underlie disease progression and resistance. And furthermore, if we do this at the single cell level, we can study the molecular heterogeneity of prostate cancer. And so using this IFD, the CTC-iChip device, we were able to isolate single cells from patients. Actually, the cells are in a tube, and we labeled them with antibodies directed against an epithelial marker. EpCAM is a mesenchymal marker, CDH11, counterstaining for any contaminating leukocytes using CD45. And we actually used brute force, so manual single-cell micromanipulation using microcapillary tubes to isolate these single cells, and then subjected them to previously established single-cell RNA sequencing methods. And to take you through the workflow, we started with 22 patients with prostate cancer, the majority of them with metastatic castrate-resistant disease, from which we isolated 221 single putative CTCs. Only 60% of these were successfully amplified in sequencing, implying that many of these had degraded RNA, or perhaps were in the circulation but were actually not really alive. And then through the application of some very stringent thresholds and criteria, most specifically, or most importantly, looking at the expression profiles, we identified those cells that had high expression of prostate lineage-specific markers, but low expression of leukocyte markers. And we ended up with 77 single prostate CTCs that we were confident were in fact lineage-confirmed CTCs. We then compared those to prostate cancer cell lines, as well as primary prostate tumors that we also sequenced. And this is the heat map. I'm not going to take you through all of it, but this is a heat map showing the unsupervised hierarchical clustering. I think the most important thing to note on this is that these colored bars here represent individual patients, and it's interesting that the CTCs from a given patient tends to cluster with one another. And if you quantitate heterogeneity, you can see that cells from within a given cell line tends to have a certain level of heterogeneity, which increases when compared across multiple different cell lines. But when you look at CTCs from within any given patient actually has the same level of heterogeneity as single cells from across multiple different cell lines. And in fact, this heterogeneity increases further when you look at CTCs across multiple patients. And if you look at this with a little more granularity, looking at individual genes that are expressed, as expected, the CTCs do express prostate lineage-specific markers. A little bit surprisingly, we saw greater expression of epithelial markers compared to mesenchymal markers in these CTCs, and there's great heterogeneity in terms of stem cell markers and proliferation markers. So we then looked at what genes and pathways are enriched in prostate CTCs compared to primary tumors, and we saw that 711 genes were specifically enriched in CTCs compared to primary tumors. And when we compared that to existing data sets of metastatic tumors and as well as to primary tumors, we ended up with 21 pathways that were specifically enriched in CTCs compared to primary tumors and compared to metastases. And many of these were involved in, for example, growth factor signaling, hormone signaling, and cell adhesion signaling. We then looked at the clinical question of what are potential mechanisms of resistance to enzalutamide, this potent anti-androgen, in metastatic prostate cancer. And to this, we performed a retrospective analysis of the data we had at hand, which included 36 CTCs from patients with enzalutamide resistance and 41 CTCs from patients who are naive to enzalutamide. And using gene set enrichment analysis, we were very surprised to find that the non-canonical Wnt signaling pathway was enriched in the enzalutamide resistant compared to naive patients. And this is the non-beta-catenin dependent branch of the Wnt signaling pathway, and traditionally thought to be involved in cell polarity, movement, cytoskeletal reorganization involving RORAC CDC42. This was a bit of a surprise. There are other proposed mechanisms in the literature at the time, so we looked at these as well. So, for example, androgen receptor splice variants have been in the news recently, and specifically ARV7, which has a truncation of the ligand binding domain of androgen receptor and is thus constitutively active. In this particular data set, we did not see a significant enrichment in the resistant versus naive population, although we did see increase in these variants compared to primary tumors. Part of the reason for this may be that, again, this is a retrospective analysis. These patients happened to also receive, many of them had also happened to receive abiraterone already. So that perhaps explains that result or lack of result. But I think even more striking than that is really the striking heterogeneity that we see in terms of androgen receptor splice variants. Each of these numbers represents a different patient, but even within a different individual patient, you have some cells that express it, some others that don't, and some of these cells actually express multiple different AR splice variants, even within the same given patient. So there's really striking heterogeneity. We also, other study wasn't designed to do this. We looked at, we were able to pick up some AR mutations, but again, not significantly enriched. And then using a different bioinformatic method, we also, again, looked at non-canonical Wnt signature, which again was highly enriched in enzalutamide resistant compared to naive patients. We also looked at a previously published mouse xenograft model and did bioinformatic analysis on this. This model was from Charles Sawyer's lab, and it was actually a model for enzalutamide resistance that was derived from LNCAP cells. And this model also showed enrichment of the non-canonical Wnt pathway in the enzalutamide resistant compared to control xenografts. And even more interestingly, this particular paper actually proposed glucocorticoid receptor as a potential mechanism for enzalutamide resistance. We saw that when we stratified these xenografts by GR expression, we saw that those models that had GR high expression actually no longer had a significant enrichment of non-canonical Wnt, whereas those with GR low expression had a significant enrichment of non-canonical Wnt. And this was reflected again in our own CTC data set as well. And we looked in vitro when we overexpressed non-canonical Wnt ligands. Of all the non-canonical Wnt ligands, Wnt-5A conferred the greatest level of enzalutamide resistance in vitro. And when we We noted also that when we just simply treat LNCAP cells, which is the prostate cancer cell line, with enzalutamide, we see increased induction of Wnt-5A expression, and when we suppress Wnt-5A, then we restore sensitivity to enzalutamide. And through an orthogonal approach, we also looked for expression of Wnt-5A in CTCs and tumors using RNA in situ hybridization, confirming the expression of this ligand in a subset of tumors cells. So in summary, we were able to obtain high-quality single-cell transcriptome profiles of CTCs from patients with prostate cancer. We noted considerable heterogeneity in single CTCs, both within patients as well as across multiple patients, within individual patients. We noted multiple different AR splice variants, even within individual CTCs from a given patient. We identified non-canonical Wnt signaling as a potential mechanism of resistance to anti-androgen therapy, and really heterogeneous signaling pathways may contribute to resistance. This study is clearly limited by the small number of patients, relatively small number of CTCs, due to the technical difficulties of obtaining these cells, retrospective nature of the analysis, and large trials are necessary to confirm these findings, but I do think molecular analysis of CTCs is promising for identification of drug resistance mechanisms and as potential biomarkers. And I'd like to thank Drs. Daniel Haber, Shamla Maheswaran, Mehmet Toner, who are my laboratory mentors, as well as funding sources, and thank you all for your attention.
Bruce. I have a question. So, did any of these patients who had splice variants and other evidence of resistance when they started therapy, did any of them actually respond to treatment?
So the question was, did any of these patients have these aberrations prior to the start of therapy?
They had them prior. You showed that they did, but did any of them actually respond to treatment in spite of having the splice variants or non-canonical pathway activation?
So there are some patients who had some expression in some of the cells, and so it's not clear-cut black and white. You have it and you don't respond. So I think there's definitely great complexity here that we need to deconvolve.
So you really need a larger series of patients to figure out whether these changes are really conferring resistance.
Yes.